I'm going to talk about, as the title says, stem cells and genomics from precision medicine to clinical trial in a dish. And again, AJ, kudos to doing such a fantastic job with the Journal of Cardiovascular Aging. And excellent. Let me just take it back 1 second. Let me just take it back to the very beginning. Let me point out. So you can see this, right? Okay.
Yes.
Yeah, so this is my disclosure slide right here. And before I start, I just want to let everybody know about the AHA's second century vision. I think as some of you may know, this year is our 100-year celebration. I think it will occur on June 10th, 2024. And whether you're a basic scientist or a translational researcher or a clinical investigator, I hope you embrace the 2024 American Heart Association's second century vision. And that is, we want to advance health and hope for everyone and everywhere. I think what sets the AHA apart is our commitment to funding research. For example, last year, we raised about $1.1 billion and provided $180 million of funding. We are the largest funder of cardiovascular science in the U.S. outside of the National Institutes of Health. Since 1949, the AHA has funded more than $5.7 billion. And currently, we have 1,700 active awards in our portfolio that ranges from basic to translational to clinical to population science. And besides that, what some of you may not know is that AHA also work across more than 100 different countries and 130 sister societies. For example, for the Go Red for Women, we have programs and partnerships with 38 organizations in 51 countries. And part of my job is to be a global ambassador for the AHA. For example, last October, I was at the Great Wall International Congress of Cardiology meeting, along with several of my colleagues at the from the AHA BCBS Council. I was fortunate to meet with the U.S. Ambassador to China, Nick Burns, along with the AHA officer John Miners, in which we discussed some of the issues that pertain to the U.S. and China, as well as broader collaboration in biomedical science. So here are some ways that you can help. For the help the AHA, you can volunteer for local AHA events such as Heart Walk, Heart Ball, or Go Red for Women luncheons. You can become involved with local board or committees. You can also sign up to be a You're the Cure advocate. You can be an AHA spokesperson for events, missions, programs, and fundraising efforts. And here's the link if you want additional information to be a volunteer. And so thank you so much for those of you who are members of the AHA and who are supporting the AHA. So with that, I just want to start the talk by giving the audience here a brief clinical case report that I saw at Stanford and how it ties together stem cells, genomics, drug discovery, and precision medicine going from precision medicine to clinical trial in a dish. And so this is a 57-year man, previously healthy, comes to the clinic with newly diagnosed non-ischemic cardiomyopathy, found to have severely reduced left ventricular ejection fraction and family history remarkable for multiple sudden cardiac deaths, premature AFib, heart failure, hypertension, prompting a clinical genetic testing. Genetic test shows a pathogenic variant in lamin AC. The question is, how does this genetic information affect the management of his condition? As many of you know, there's a significant interest in the Precision Medicine Initiative. This concept was first promoted by President Obama back in 2015. And much of this is made possible by recent advances in next generation sequencing, bioinformatics, and big data. So by understanding our genes along with lifestyle and environment, the hope is that we can better predict which treatments might work best for us. Now, in my opinion, I think there are 3 key technological breakthroughs that marries precision medicine and basic science research. The first one is the DNA sequencing, and I think as y'all of you know, the technology has become much cheaper with lower error rate, broader coverage, and much faster turnaround. By back in 2000, it cost $1 billion in over 13 years to get the DNA sequence. Today, you can get the DNA sequence for less than $500 with turnaround time of about 3 to 4 weeks. And with increasing usage, the idea is that clinical care could be more predictive, preventive, personalized, and participatory. In my opinion, the second key technological breakthrough is the various types of genome editing technologies, starting first with the zinc finger nucleases, then TALEN, then culminating with the CRISPR-Cas9 discovered by Emmanuel Charpentier and Jennifer Doudna. So using these molecular scissors, you can now change the DNA of animals, plants, microorganisms, and stem cells. And it's a very fast-changing field as there are new types of editing technologies that have become much more efficient and much more targeted, and hence resulting multiple startup companies that are being formed in this space. In my opinion, the third key technological breakthrough is the human iPSL platform. This provides you then with a test bed to validate human mutations from your sequencing, to modify genetic variants from your CRISPR editing. And I think, as you know, the technology was pioneered by Dr. Shinya Yamanaka in 2006, led him to receive the Nobel Prize in 2012. And just like human ESLs, these iPSLs can then be differentiated into any somatic cell types, such as cardiomyocytes, neural cells, hepatic cells, islet cells. This then allows you to characterize the disease phenotype, identify the molecular targets, and implement personalized precision medicine. And so over the past 10 years or so, we at Stanford, in collaboration with Greenstone Biosciences, we have made iPSLs for more than 2,000 patients with different medical history, ethnicity, and sex. We use PharmGK to help us understand how human genetic variation impacts the drug response phenotypes. These cell lines are linked to de-identified medical information using the clinical database. We work with FDA on drug safety testing, and we have a very robust sharing resource plan with the broader scientific community, which I'll explain later. This is our typical workflow. We recruit patients from diverse populations. We generate the 2D cardiomyocytes, 3D human cardiac organoids, and also 3D engineered heart tissues for disease modeling and also for drug screening. Now, with these three tools of genetics, genome editing, iPSLs, you should be able to study cardiovascular genetics and epigenetics. As many of you know, our knowledge of genetics and epigenetics has increased dramatically over the past decade. For example, we now know that there are different types of genetic changes, such as single nucleotide polymorphism, copy number variation, and also chromosomal abnormalities. Likewise, there are many different types of epigenetic changes, such as histone modification, DNA methylation, and RNA, non-coding RNA. These changes then play important roles in coronary artery disease, cardiac arrhythmia, cardiomyopathy, which I'll highlight in some of the studies that we've done, aortopathy, dyslipidemia, and also pharmacogenomics, which I'll highlight in my talk as well. So this is one of the earliest examples of using iPSLs. This was published back in 2012. Here we model familial dilated cardiomyopathy. This is a 3-generational family with DCM. The boy here already had a heart transplant at age 15, and the mutation is due to troponin T with arginine to tryptophan switch at codon 173. And compared to the healthy control cardiomyocytes, we found that the DCM cardiomyocytes had altered regulation of the calcium ion, decreased contractility, and normal distribution of the sarcomere alpha-actinin. And importantly, you can treat these cells with a drug such as beta blocker, metoprolol, to improve the function of these cells, recapitulating results from multiple large beta blocker clinical studies. This is a follow-up study we published a year later, in which we were referred a large family with a familial hypertrophic cardiomyopathy due to myosin heavy chain 7 mutation with arginine to histidine switch at codon 663, The mother here had HCM with symptoms of cardiac arrhythmia. She has 8 kids. The 2 oldest already manifest the HCM phenotype. Kid number 3 and 8 were genotype positive, but phenotype negative, most likely because they were still young at that time. And then through a whole series of experiments, we're able to show that this particular mutation causes increased intracellular calcium, which leads to calcium handling defects, and then cellular arrhythmia, also activates the calcineurin and fat signaling pathway, which causes hypertrophic responses. And importantly, since you set this up as a platform, then you can then treat these cells with calcium channel blockers such as verapamil or a beta blocker such as propranolol to block the arrhythmia and block the cellular hypertrophy. Now, in pediatric population, the third most common type of cardiomyopathy is left ventricular non-compaction or LVNC. This occurs in about 1 in 1,000 patients. In this study, we were for a large family cohort from Stanford and a small family cohort from Japan. The boy here already had a heart transplant, age 5. Older brother and older sister also had LVNC, but were asymptomatic. And then through a whole series of experiments, we're able to confirm the mutations in cardiac transcription factor TBX20, as shown here, 1 in the T-box domain, and then for the Stanford boy, and then 1 in the transactivating domain for the Japanese girl, causes abnormal activation of TGF beta signaling pathway, which recapitulates a key aspect of the LVNC pathophysiology. Now, what I've shown you so far are 3 examples to model Mendelian mutations, such as troponin T in DCM, myosin heavy chain 7 in HCM, and TBX20 LVNC. In addition, these cells can also be used to study the impact of single nucleotide polymorphism with SNP. And one of the most prevalent SNP is this aldehyde dehydrogenase genetic polymorphism, which causes what we call the Asian flush syndrome. This deficiency occurs in about 36% among East Asians, 8% of the world population. Missing this enzyme is associated with increasing risk of heart attack, increasing risk of a higher incidence of hypertension, cancer, and also increased complication from type 2 diabetes. I actually don't have this enzyme, so we've been studying this for a while. And so this enzyme deficiency has been well documented to be a risk factor, again, for myocardial infarction, Japanese men, in Korean men, and also in Han Chinese. And so for this study published about 10 years ago, we recruited 10 Stanford undergrads. 5 of them are wild type, 5 of them are heterozygous. And heterozygous means that they have the facial flushing. If you're a homozygous mutant, which is about 1% of the population, the symptoms get very severe. And here we model myocardial infarction by giving the cells a hypoxia. And as you can see here, the wild type tolerated hypoxia quite well with minimal apoptosis, whereas the heterozygous has much more apoptosis with the same amount of hypoxia. So overall, we believe the enzyme is involved not only in detoxifying alcohol, but also getting rid of ROS. And after a heart attack, there'll be tons of ROS that gets activated. And if you're missing this enzyme, you have a harder time getting rid of ROS, and hence leading to more cell death and perhaps explaining for the worst clinical outcome observed in epidemiology studies. Now, in a more recent follow-up study from last year, we then used the iPSC-derived endothelial cells from these patients and exposed them to alcohol to model vascular dysfunction and coronary artery disease. We know from large genome-wide association study using the Biobank Japan data set that this deficiency is one of the strongest SNP-associated coronary artery disease. And mechanistically, this is because alcohol is broken down to acetaldehyde, which is toxic and causes damage to the whole vascular system, increases ROS, increases vascular inflammation. Acetaldehyde is further broken down into acetic acid by aldehyde dehydrogenase, and acetic acid is non-toxic. So if you're missing aldehyde dehydrogenase, you have more acetaldehyde in your systemic circulation, which causes the blood vessels to vasodilate, which causes your face to turn red. It also causes chronic usage, causes alcoholic cardiomyopathy, post-holiday heart syndrome, alcohol-induced atrial fibrillation, and so forth. And here, what we did is we did a compound library screen and also molecular docking analysis to show that a drug, SGLT2 inhibitor, empagliflozin, can actually mitigate the vascular dysfunction. And empagliflozin does this by alleviating the ALDH induced oxidative stress by inhibiting the sodium hydrogen antiporter 1 and also activating the AKT-ENOS pathways. We also confirmed these findings in 2 different mouse models of diabetes and alcohol exposure, showing that empagliflozin can ameliorate these vascular dysfunctions. So I think these are examples of 2 papers using the iPSL to study alcohol intolerance among East Asians. And what I'm trying to highlight here is that you should be able to use this model to study the genetic susceptibility among different ethnicities. For example, why African Americans have much more hypertension, Hispanics have much more diabetic vasculopathy, and South Asians have much more metabolic syndrome. Now, one of the most commonly asked questions that we get asked is, how do you decide then between 2D versus a 3D? The simple answer is that there's no single size that fits all. For example, if you want high throughput with 2D, then you sacrifice tissue maturation. On the other hand, if you want high tissue maturation, then you sacrifice throughput because it's more costly to generate. And then similarly, there are many obstacles and opportunities that you still need to overcome for the 3D model, including vascularization, extracellular matrix mechanics, optimization, universal cell type media, especially for microphysiological systems, scaling up for industrial applications, and integration with other organ-on-chip system. But I do think for the young investigators in the audience, this is a very fertile area of investigation, and you'll see a lot of people working on this in the next few years. And for example, here's a recent paper that we published using 3D human engineered heart tissues, the model tachycardia-induced cardiomyopathy. And this is in collaboration with Dr. Luisa Mestroni and also with Matthew Taylor. And here, this tachycardia-induced cardiomyopathy is due to, for example, rapid beat from the atria, like atrial fibrillation, atrial flutter, supraventricular tachycardia, a rapid beat from the ventricle, such as idiopathic LV tachycardia or idiopathic RV tachycardia. I think in either case, the abnormal calcium handling and the reduced cellular metabolism and the inefficient energy usage can cause this contractile dysfunction as outlined in this cartoon here. So I want to switch gear to the second part of my talk, which is using how we use these iPSLs to drug screen. And I think, as you know, this process is time-consuming, expensive. For example, typically costs about $1 to $2 billion and 10 to 15 years to bring a new drug from discovery to clinical trial to market approval. And even worse, many of the drugs that you take today actually do not work at all. And so shown here are the top 10 sales for drugs in the US. Blue means the drug works, and red means the drug does not work. And so for Abilify, which is used to treat schizophrenia, out of 5 people, it helps 1 person. With Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. So overall, this is pretty poor response rate, and this is the reason why the editorial here is titled Time for 1 Person Clinical Trial. Now, in the US, both the FDA and Big Pharma are also thinking about using iPSLs to accelerate the drug development process. This is a study published by the FDA and several companies in which they use human iPSLs to predict arrhythmia as part of the Comprehensive In Vitro Proarrhythmia Assay, or CPA initiative. And I think, as you all know, all the drugs need to be tested for cardiac arrhythmia. And currently, we test them on using Chinese hamster ovary cells or on human embryonic kidney cells that overexpress the hERG potassium channel. Not ideal because the children HEC cells are obviously not cardiomyocytes. And here, the investigators use 1 consensus protocol, 2 human iPS lines, 3 continents, 28 blinded drugs, 2 statistical models, and 10 volunteer labs. Importantly, they were able to show an 87% prediction rate. This is very good considering that this is one of the earliest studies by the FDA CPEC group. And that means there's going to be significant room for improvement in the future. Now, in our lab, we've been pushing that direction as well. This is a paper we published in 2016 using transcriptomic profiling iPSC-derived cardiomyocytes to predict individual drug safety and efficacy responses in vitro. And here we tested 2 drugs, rosiglitazone and tacrolimus. We also use toxicology analysis, CRISPR genome editing to evaluate and confirm the potential mechanisms of action, especially for rosiglitazone. This drug was actually withdrawn from the market in 2000 because of increased cardiovascular risk. And we developed algorithms to help us predict the drug responses. And so one of the goals in my lab is to create what we call a human encyclopedia of transcriptomic signatures of drug responses on heart tissues. So shown here are 2 examples whereby we assess the effects of 4 different kinds of calcium channel blocker, nifedipine, amlodipine, diltiazem, and verapamil, 4 different kinds of statins, atorvastatin, lovastatin, simvastatin, fluvastatin, and we assess them using human iPSC cardiomyocytes or human iPSC engineered heart tissues. And so what's interesting is that you'll see different gene expression patterns among these different drugs, even though they belong to the same class of drugs, they may actually exert very different effects among different individuals. And so for the statin story, this is a follow-up study in which we use iPSCs and multi-omics to dig deeper into the role of statins in vascular health. I think some of you may know statins are given to over 40 million adults in the US, including myself. I actually take Lipitor, atorvastatin. My primary care doctor just check LDL to see how low it goes. But what is the effect of statin on a person's heart tissues, on a person's vascular tissues, on a person's brain tissues, and so forth? And so there's a lot we still don't know in terms of the biological and beneficial effects beyond cholesterol lowering. And here, using ATAC-seq, we observe changes in gene accessibility in the human iPSC-derived endothelial cells when they were treated with simvastatin. We found that the simvastatin can protect cells from endothelial dysfunction by epigenetic regulation of the YAP pathways, which control genes that are involved in the endothelial to mesenchymal transition. And then finally, we validated these findings in a diabetic mouse model to confirm the in vivo protective effects of a statin. Now, besides the cardiomyocytes, these iPSCs can also be differentiated into all these different cell types. And this slide shows the differentiation of iPSCs into mesoderm by GSK inhibition, into cardiovascular progenitor cells by Wnt inhibition, into cardiomyocytes by Wnt activation, and into cardiac fibroblasts by TGF-beta inhibition, into smooth muscle cells by PDGF activation, and into endothelial cells by VEGF, as shown here. A lot of this protocol has been worked out over the past 20 years, but these are still not perfect. And again, there's going to be significant room for improvement of generating these different cell types. And this is also another fertile ground of research for the young investigators in the field. Just an example, here we develop a robust chemically defined protocol for generating cardiac fibroblasts. And as this is the protocol listed here. And since you're able to generate the cardiomyocytes and the cardiac fibroblasts from the same individual separately, you can study the crosstalk and interaction between these 2 cell types. And as one example here, you can give TGF-beta, which causes cardiac fibroblasts to undergo myofibroblast transdifferentiation and contribute to cardiac tissue fibrosis. Interestingly, the cardiomyocyte can attenuate this process by a paired mechanism through the AMP-BMP and the natriuretic peptide receptor this pathways, or NPR1 receptor pathways. And then some of you may know sacubitril. This is an active molecule that is part of the drug called Entresto, very effective for treating patients with heart failure. And mechanistically, we found that sacubitril can exert its antifibrotic effect by blocking neprilysin, which is the enzyme that degrades AMP, BMP. We also have an interest in chemotherapy drugs, and this is because heart disease and cancer account for the top two causes, morbidity and mortality. And as more cancer patients survive, they end up in the cardiology clinic because of symptoms of a heart failure. And so shown here is another study that we use a human iPS cell to predict chemotherapy-induced cardiac toxicity. Here we recruited 8 breast cancer patients. A year later, some of them had normal cardiac function, while others had doxorubicin-induced cardiomyopathy. We then generated their cardiomyocytes, exposing the doxorubicin in vitro. We then studied the cellular responses by these assays listed here. And as proof of principle, we showed that if we pick the patients appropriately, these iPS cell-derived cardiomyocytes can be used to predict who's going to respond and who's not going to respond. This is actually a nice editorial written by Dr. Tim Kemp as well. And in a follow-up study on breast cancer patients, we showed that these cells can also be used to model Herceptin-induced cardiac dysfunction. This is a very common chemotherapy drug used for breast cancer patients. Here, we recruited 2 patients who did not exhibit toxicity, 2 patients who developed moderate toxicity, 3 patients who developed severe toxicity after Herceptin. And what's interesting is that in contrast to doxorubicin, which causes sarcomere disarray and cell death, Herceptin causes more of a metabolic derangement and mitochondrial dysfunction. And then we also use chemical library screen and also able to show that metformin can be used to rescue some of the cellular phenotype. Now, besides doxorubicin, other popular chemotherapy drugs include tyrosine kinase inhibitors, or TKIs. These TKIs kill cancer cells by inhibiting the phosphorylation activity of hyperactive receptor tyrosine kinases. And common TKIs you may have heard of include Gleevec and Sutent, which inhibit VEGF-R2 and PDGFRB in cancer cells, but can cause heart failure, a side effect because both VEGF and PDGF signaling pathways are also critical regulators of cardiovascular development. And in this study, we generated iPS cells in 13 patients, treated them with 21 most common TKIs, came up with a cardiac safety index that allows us to predict which TKI is cardiotoxic and which TKI is not. So the ultimate goal is to further optimize the safety index so that if you give me a drug, I can help predict the safety profiles of the drug before giving them to patients. So as you can see from these examples, we're integrating iPS cell biology with various cellular, molecular, and functional readouts and multi-omics analysis to help us better understand the diseases at a systems biology level. This is a editorial or review article written by my former postdoc, Edward Lau, who's now a faculty at University of Colorado, Denver, Anschutz. And what we're doing now is integrating large-scale human iPS cell populations, 100 cell lines, 200 cell lines at a time, so that we can understand the EQTL, the expression quantitative trait loci, and also to study the genotype-phenotype relationships using these co-expression networks. Now, because of the fast advances in AI machine learning, I think you'll also see a lot of drug discovery-based models in the next decade. For example, working collaboration with computational scientists at Greenstone, we routinely use these AI machine learning softwares that allows us to analyze the protein structure, and then we use in silico molecular docking. Once we know the molecular target and protein structure, we can then screen for about 1 billion compounds at a time. Many of these are virtual compounds, which means they haven't been synthesized at all. And then we can then synthesize the drug hits that we want and then further validate it using iPS cell model. And so this is a schematic of our typical molecular docking campaigns at Greenstone. You can start with different sets of compound libraries, such as the Zinc 22 library, FDA repurposed library, chemical library from the enemy located in Ukraine, and the Wuxi chemical library located in China. Alternatively, you can also perform small sampling size of 100,000 compounds at a time for your docking campaign. Once you identify the docking site, you can then further refine the rank score, as well as perform molecular dynamic simulation to look for compounds with desired drug-like properties. And so here I just want to highlight one example using this AI machine learning in silico docking for drug discovery that I just mentioned. I think, as you all know, marijuana is being used increasingly in the US due to legalization. Interestingly, the UK Biobank data set shows that marijuana use is associated with increased myocardial infarction, but the exact mechanism is unknown. And here we show that the delta-9 THC, which is a psychedelic component of marijuana, can cause vascular inflammation, oxidative stress by activating the MAP kinase, NF-kappa B, and cytokine pathways. And it does so by touching the CB1 receptor, cannabinoid 1 receptor. And so we reasoned that we need to develop or come up with some kind of compound that blocks this right here. And so we use in silico chemical library screen and found that genestin, which is an isoflavone, serves as a neutral CB1 antagonist. That means it sits there. It can block binding, competitively block binding of the THC from binding here. But it's because it's a neutral antagonist. It sits there, it doesn't activate it. And so this is actually also found in soybean products. And so we are in the process of further modifying this molecular structure that enhances that bioavailability and in the process of doing DMPK and CMC studies. For the last part of my talk, I just want to touch upon how we can then use these cells to implement the concept of clinical trial in a dish. Here, the concept is that we should be able to recruit patients, generate their cell types of interest, test which patients respond to drugs and which patients do not by using their cells as surrogate, and then go back to perform the actual clinical trial on patients who are positive responders. And that way you have a higher chance of success rate for your phase 1-2 clinical trials, decrease the cost, and accelerate the timeline of drug development. So this concept is timely because some of you may know the FDA Modernization Act 2.0. This was recently passed by both the Senate and the House on December 2022. The law authorizes the FDA to use alternative testing platforms such as stem cells, organoids, and in silico modeling. And then the goal here is to improve the predictiveness of preclinical study. And also the U.S. Environmental Protection Agency, or EPA, will completely eliminate all animal testing by 2035. This slide highlights, in my opinion, what I think is one of the major problems of high failure rate in drug discovery. That is the lack of genetic diversity and pharmacogenomic differences with traditional models, such as using immortalized cell line in the inbred mouse. For example, because these are immortalized lines, or the differences that you see here between vehicle versus drug is actually due to a technician's pipetting skill. Likewise, the differences that you see here, because these are all inbred mice, as 1,000 FAB mice, essentially the same thing as 1,000 Joe Wu, for example. The differences that you see here is actually due to a technician's tail vein injection skill. On the other hand, the differences that you see here in a population of, say, 1,000 different diverse patients, is actually due to their genetic variability that exists among all of us. And this pharmacogenomic difference is ultimately what you're trying to understand before you go into clinical studies. And this is otherwise impossible to capture or recapitulate using traditional immortalized cell line inbred mouse cell models. So I'm on the FDA Cell Tissue and Gene Therapy Advisory Panel. That means we review the phase 3 trials before they get approved by the FDA. And this slide is given to me by my colleagues in the FDA. So for rare orphan disease, the FDA recognizes the challenges of developing new drugs due to small number of patients, and yet there are thousands of genetic variants for each disease. And so now their approach is to accept data generated from cell models to test drug efficacy. As an example, for cystic fibrosis, the drug Callidico, made by Vertex Pharmaceuticals, initially approved for 10 genetic variants, and now expanded to 24 more genetic variants. Another drug is Fabry's disease, lysosomal storage disease, passed on by the X chromosome. The drug Galafold, made by Amicus Therapeutics, initially approved for 40 genetic variants, now approved, expanded to 348 genetic variants. And currently, the FDA is interested in expanding the drug-based, cell-based platform across the full spectrum of diseases. And this slide is also given to me by the FDA. I think the value proposition here is using these in vitro efficacy data to reduce the number of clinical studies to inform the regulatory decision-making at the time of marketing application, to streamline the drug development process, and also to expand the indications based on in vitro data. So let's come back to this patient that we're seeing at Stanford, 57-year-old man who have non-ischemic cardiomyopathy. Turns out the patient belongs to a large family cohort with a Lamin frame shift mutation that leads to early termination. So Lamin cardiomyopathy accounts for about 10% of familial dilated cardiomyopathy. And in this large family, as you can see, many of the individuals had cardiomyopathy, atrial fibrillation, and atrial ventricular block. And importantly, eight of them died from sudden cardiac death. And to make a long story short, we figure out the mechanism is due to abnormal activation of the PDGF signaling pathway, which then leads to abnormal cellular arrhythmia and also impaired contractility. We also showed that by blocking the PDGF pathway with the PDGF inhibitor, quinolonide at a low dose can rescue the abnormal phenotype of these cardiomyocytes. However, quinolonide is not an FDA-approved drug. It's a phase 3 trial used for cancer. So we can't really give quinolonide to our patients. But what's interesting is that we notice many of these young patients develop early onset hypertension. And this is kind of weird because usually when you have cardiomyopathy, your ejection fraction is low. And so we decided to study this by comparing the endothelial cells in the Lamin patient versus the control healthy patient. And we did a whole bunch of assays as shown here. And we showed that KRF2 as a transcription factor may be responsible for the endothelial dysfunction that we saw in the Lamin iPSC-derived endothelial cells. And this time we used an FDA-approved drug library screen, and we found lovastatin can be used to activate the KRF2, as shown on this cartoon here. And then we then decided to give lovastatin back to the Lamin patient, and hence the title here is Clinical Trial in a Dish using iPSC to show lovastatin can improve endothelial dysfunction and cellular crosstalk. So these same patients, when we treated their cells, were given lovastatin daily. Remember, this is to treat the vascular dysfunction, not to treat the LDL. And then the clinical data is shown here. We use EndoPad, which is a machine that can measure flow-mediated dilatation during reactive hyperemia. Essentially, it's a blood pressure cuff that occludes your arm for about 5 minutes and releases it, and it's trying to measure how much blood flow come back. Normal index is 1.67. Abnormal index is less than 1.67. You can see here this person started with a very poor index of 0.48, and then after lovastatin treatment improved to 1.06. So I think this is what we call a clear example of clinical trial in a dish, whereby we first identify a large family with Lamin mutation, very rare, elucidated the mechanism of the cardiac as well as the vascular dysfunction, screened for a panel of FDA-approved drugs using the iPSC-derived cells, showed that lovastatin can work, and give the lovastatin back to the same patient here. And so I really believe that the FDA Modernization Act 2.0 will pave the way for the next era of drug discovery that uses predictive AI, generative AI to find the optimal candidate, similar to the story that I showed you earlier involving Genestin as a neutral CB1 antagonist to mitigate marijuana-induced vascular toxicity. And as shown here, these drugs could also be peptides, antisense oligonucleotides, RNA therapeutics. And then you use the traditional in vitro and animal studies of your PK/PD But what we're proposing is that before you go into patients, you can implement this clinical trial in the dish here to help you predict the safe numbers of patients. It's almost like a sneak preview before you go into a clinical study. So in summary, I believe there'll be a paradigm shift in the future using these patient-specific cells to help us understand the molecular mechanism of diseases, along with multi-omics, genomics. This would then create disease-specific data sets, which then feeds into AI machine learning for molecular docking, molecular dynamic simulation, and predictions. We can then use these drug targets to come up with drugs that are more precise. We can then test these drugs on the iPS cells in a dish, and we can then acquire additional data, refine our AI machine learning, refine our drugs, and goes around the cycle for many, many different iterations. So this concludes my talk. I want to acknowledge my postdocs and my instructors, as well as my former trainees whose work I highlighted here, my collaborators at Stanford and outside of Stanford, and also funding support as listed here. And I think for those of you who is interested in drug discovery, we host an annual drug discovery symposium. It will be coming up April 29th to 30th. The meeting will be both in person and also by Zoom. We typically have about 600 in person and about 6,000 virtual attendees. The speakers are usually CEO, CSO, CMO, big pharma, and biotechs, NIH, FDA, for example, Dr. Rob Califf, and then Peter Marks, the director of CBER, Center for Biologics, and then Patricia Cavazzoni, the director of CDER at the Center for Drug Evaluation. The goal here is really to have intimate discussion about what's working, what's not working, how do we improve, how do we improve the process so that we can come up with better therapies that are safer, faster, and cheaper. So the ultimate goal is to help our patients. And then lastly, for those of you who are interested in these cell lines, you're welcome to reach out to Greenstone Biosciences. They provide cell lines free of cost to academia institutions. You just pay for the shipping costs. And we hope this will encourage more people to use this powerful platform to study human diseases and listed some of the cardiac diseases as well as non-cardiac diseases as well as rare orphan diseases. And I'll stop here. Thank you very much for your attention. And again, thank you, AJ, for inviting me here. And kudos to a great job with the journal.